Dr. Chiocca?
Yes, hi. Congratulations, Dr. Mendell and group, for a nice preclinical study and a nice protocol for this disease that devastates neonates. Clearly, this is something that should be brought forward. I sent him some questions, and I know you answered them, and I'd like to go through each one of them. My first question included the fact that in your exclusion criteria, you're excluding patients who are taking drugs for myopathy, neuropathy, or antidiabetics. And I think you answered it fairly satisfactorily, explaining why, if they were taking valproic acid or sodium phenylbutyrate, you wanted to exclude them. And I also agree with your exclusion criteria for patients that are diabetic. The question is, just one additional point of clarification, though, relates to what happens if you have a patient that was taking valproic acid or sodium phenylbutyrate as part of a clinical trial just on their own. Will you allow them to go on the trial after a certain period of time, or are they completely excluded?
I think in the first small sample group, we would prefer not to have any potential for carryover effect. We'd like to be able to have this as clean as possible. Hopefully we will have efficacy, and we would not, like our critics, I guess, to imply that there were other factors other than gene delivery. So we would reserve that group. This is a very small sample, and God knows if we're successful, we could extend this to many patient groups. So I think we would stick to that point, unless you had unless you advised us differently or thought about it differently. But that's how I think we're thinking about it. John, you want to make a comment?
I'll just make one comment.
Why don't you come up here?
Yeah, Nino, this is John Kissel, and I'd like to just make one other comment in addition to what Jerry said. And since we have a number of lay people, family members here today, that I agree 100% with what Jerry said for the reasons he said. I think it is worth mentioning, though, that there's now four trials that have been done with valproic acid. There's one that's in press in adults, and there's another one being prepared in type 1 infants that really showed no efficacy. So I think, as Jerry said, if any efficacy is shown in these preliminary studies, I think the fact that they have been on valproic acid will not be a hindrance to expanding this. And I think that's very important for families to understand, because we get asked that all the time, because valproic acid is available by your local pediatrician.
But you wouldn't. But as far as inclusion for the first group.
Exactly. But right now, I agree that you would want to keep it clean.
And I certainly would agree with your rationale. It sounds like for the first trial, that probably you should have a very clean group. Thank you for answering. The second question had to do with the fact that your last cohort was not expanded to 6 as per usual routine dose escalation trials, and that you kept it to 3. And you answered that this really had to do with vector production issues and current production methods, and that this really restricts the enrollment. And I certainly can understand that. I think I'll come back to this issue towards the end again. But at this point, there's no need to discuss this any further. And the next issue was, again, similar. Why not increase dosing to a maximum achievable dosing if possible? Why stop at 3.3 times 10 to 14 vector genomes per kilogram? And again, you explained that this is your maximum achievable dose, and that this is your limit, and that and I do understand the answer to that question. You discussed, and I was uncertain and unclear on the fact of why patients that had only one copy of SMN2 were excluded from the trial since this was just a safety trial. And you gave the rationale of the fact that these patients died so fast before 1 month of age that it probably would be very difficult to enroll them and perform a safety study on them. And I think that was very well explained in your slideshow. So I really have no, I'm in favor of your argument. Again, you in your trial, your protocol, you explained fairly, very well, and very rationally, and very logically why you're targeting children rather than adults. And I think your survival curves showing how long adults can live with this disease really illustrate the fact that in a trial like this, it would be very hard to look at safety and efficacy with a population with the more benign forms of SMA that live very long. You had a typo on page 10, which you have fixed, and therefore there's nothing else to discuss there. Again, I agree that your efficacy and safety data are supported and justify the proposed trial. My 8th question had to do with the justification for the starting dose in humans based on the safety data in mice and primates, and how you came to the dose in humans, and it seems to me that the starting dose in humans was low. I think both in your presentation as well as in your answer to me, you provided your survival curve showing the exquisite importance of dosing in providing efficacy, and I think that that's also clarifies the issue. The 9th question had to do just with another typographic error on the use of electrical impedance myography, and I accept and I thank you for the clarification. The 10th question had to do with your preclinical data that excluded subjects with antibodies to AAV9, and again, both your presentation as well as the table you provided. Answer that question in a satisfactory manner for myself. So the last important question I had to do with the fact that you were going to use just one grade 3 or 2 grade 2 toxicity as becoming stopping rules for the trial. And my main concern with that is that it seemed to me that this may be really too harsh of a rule, especially for a patient population that dies of this disease and dies fairly fast. And my worry, my concern is the fact that with a dose that is with a therapeutic dose that's so narrow, my worry, my concern is that if you just had 2 grade 2 toxicities, you would be stopping this trial perhaps prematurely before reaching efficacy. And therefore, it seems to me that you agreed with this approach and that this may be something you'll permit in the IND, and I would be very supportive in trying to changing this to what's more typical, that is, if you have 2 grade 3s, expanding the cohort to 6, but I understand you cannot expand the cohort to 6, or eliminating the 2 grade 2 toxicities as being stopping rules. Again, my worry and my concern is that you will be stopping at a fairly early stage if that happened.
Well, we agree with that entirely, and that was a response to our last IND when we went in with grade 3, and the FDA came back, the clinical reviewer came back and said it had to be grade 2 before they would accept it. So we were following that paradigm, and but I agree. I think we have a very strong argument here, and you made it for us, that we'll go in with grade 3 and maybe just give a stronger rationale when we go in with the IND. And I actually appreciate your support for that.
And again, this is a patient population that seems to me that does not survive the disease. They perish fairly fast, and again, you have a very narrow dose of efficacy, and it seems to me that you really have to show that dose. You have to show that dose is safe, but again, you don't want to terminate prematurely. So I'd be very supportive of that.
Thank you.
And my last question just had to do with, again, the absolute stopping rule is similar to what we just discussed. Otherwise, I was very impressed by this trial. Thank you.
Thank you.
Dr. Chiocca, any outstanding issues?
Not to me. I think the only outstanding issue is, again, how to better define the stopping rules and what are toxicities that will make you stop.
Yeah.
Well, we'll follow your advice here, clearly.
As a RAC, we should basically bring that forward so that when they discuss this with the FDA, they will have some ability to show that others also agree.
Yep, I totally agree with you. Let's move on to the third reviewer, Dr. Zoloth.
So I too concur that this is a very elegant and optimistic idea, and for a dreadful disease. And I wish you well. I turned my attention to the consent form because, in the face of this elegance and optimism, I wanted to make sure that the risks and benefits were carefully weighed. There was an obvious problem on the consent form, which I think, of course, it's not your problem. It seems to me to be an endemic problem to a children's hospital pediatric IRB that doesn't alter the consent form. So it sounds as if it's written for a thoughtful 35-year-old lawyer and not for people with a child who's been just diagnosed with a fatal disease and need to have at least those needs and that language carefully done. And when you responded to my concern about the language, you said, you put this phrase on top, the words "you" and "your" are used in the consent form, but they refer to the study volunteer, whether an adult or child. And I just think you need to do better than this before the study goes forward. And if you need our backing to go back to your IRB and say, you're a pediatric hospital, you need a pediatric consent form that's directed at parents. I realize it has nothing to do with the particularity of your study, but it does seem, with this extremely vulnerable population that's just gotten the worst news of their lives about their brand new baby, I want everything done with extraordinary care because we want this to work and we want this to be accepted. So that's my strong
We agree entirely,
recommendation.
and I think that our IRB is fairly receptive, and if you put it as a recommendation, I don't think we'd have a problem.
Okay. I did make a comment about making sure that the anticipated lifespan of the patient was on the first page, so that was clear, and also about the unknowability of the research itself. Again, because this is a very vulnerable population with every reason to trust you and go forward, I want them to be aware of the deep unknowability in phase 1 clinical trials. And you referred me to page 9 of the consent form under section 5, but the risks there were, I think, generic, and the final risk of complete unknowability on some level, I think, should be moved to the first part, that this is a research trial that is that will involve them and their baby taking them into extremely unknown territory, and that's what they're signing up for, not these smaller risks.
I don't think we'd have a problem with that at all.
Okay. On another comment I made was I had mentioned that this was a safety trial, and I'm hearing in the written papers that you gave us and now in the review about the depth of your optimism. In fact, you say to me, however, we disagree with the comment the trial does not offer the possibility of direct benefit to the child as evidence from the data in the preclinical studies performed in mice and non-human primates. I hear that you feel optimistic about it, and the other researchers are, all of us saw that data and it looked extremely powerful, which, by the way, would be important for parents to know about the deep optimism. However, all the language of the consent form is, this is just a safety trial, which gets you exactly into the dilemma that we just talked about earlier, which is, if it's just a safety trial, then it's appropriate to go very carefully and see, is this intervention safe in infants, just like it was safe in mice? and to explore the differences between the mice brain and the infant brain would be a safety question. But on the other hand, there's an urgency to giving the highest possible dose, which would make it seem like the safety is only for the highest possible dose and not for the escalated dose, because you're convinced that both the power of the dose and the immediacy of the timing is what's going to make this work. But the consent form is written as if, A, the patients have all the time in the world to decide, because they specifically are told they don't need to make a decision quickly, when it does seem like they would need to make a decision quickly. With the very first breath of symptoms is when you want to intervene. And two, at the highest possible dose. That's a contradiction that I think has to be thought through.
As far as the patient's not making a decision as quickly as possible, that is more our attempt to not put pressure on the patients, because we would assume that there would be another candidate who would be in line. But we try to avoid, and this is we've done in every clinical trial we've done, is to clearly avoid pressuring the patient. So that's really a very key issue, and I feel that that's appropriate to follow that path.
The reason I raise it, I completely agree with that, the undue pressure, but I'm concerned about data that the researchers and the clinical trial staff and any nurse or doctor they'd encounter has, which is that critical piece of data about if you don't give it within the 10 days in the mice, it does no good whatsoever, right?
That's true.
They all will know that and have it, and the patients need it too.
Yeah.
And it's not undue pressure, it's just sort of the truth of the design of the study. And I would really urge you to find a way to tell them the truth, acknowledging fully that they understand that that's pressure, because it is pressure. It's not like you're hyping a car or something. It's the actual truth
We can share the
of what you've got.
publications with them, and they can see, what exactly the points you're making. So they, but I mean, there is a dichotomy, I think, between saying, if you want to join the study, we better do it now. But I always like to say to patients, if it is successful, there will be there's a potential for an opportunity at a later time. That's all we can really say.
But is that true?
I don't know. We don't know. I mean, translating mouse studies to humans is something that we don't know. And Brian's treated the non-human primates as late as 90 days or later. Brian? Say it.
I think, Dr. Zoloff, you're bringing up outstanding points here, and certainly things that we have thought about and quite deeply. We have treated a 3-year-old non-human primate with a vascular-delivered AAV9 GFP and targeted motor neurons. So we know that the window still exists in larger species to target motor neurons, in what appears to be efficiently, or relatively efficiently. What I think has been very difficult to determine is we've got the window of opportunity for targeting motor neurons in a mouse. And we can get into the biology if we need to, but there is a window of opportunity for targeting motor neurons in the mouse that precipitously declines as the animal...
And for rescuing the mouse, right?
Correct. It's a very rapid mouse to try to answer the question of when would we still expect either stabilization, halting the disease progression, or improving out of the remaining motor neurons. And I think no one has the answer clearly, other than in this fast-acting mouse model where we have a 15 to 16-day window of opportunity to be asking that question.
Well, I can give you my, I guess, how to put this, my commitment, I guess, is the best way to say it. When I will do the consenting for the patients, that's clear. Mm-hmm. And I will let them know that, that there is a window of opportunity, at least we saw in the mouse. We don't know how that translates to patients, and that's the best we can do.
And that's the best you can do. But I think they need that information, because if you didn't have that information, thought you had more time for a go back, and then found out later that there was this tight window in mice,
Sure.
I would think it would be disturbing. So I want to make sure that they have the information. It's not undue pressure if the pressure is really real for this disease. It's just
And
the
we
it's just
agree
the tragic
100%.
reality.
You're right on target.
Good. I had a comment that this is so much travel and so much frequent testing that it might be quite burdensome unless you lived right near the study house, the study site. And I was glad that you used the language that I had said. Thus, the entire lifespan of your child may be that of a research subject, so that would be clarified for them. That would be involving in constant testing and constant surveillance.
And our hospital is very well equipped to handle that. I mean, we had just finished a trial where we had patients come weekly from every place in the entire country, and our human services department helps with that. We can get them into the McDonald House for unlimited periods of time. We can even help with the resources from the hospital to help pay for any problems there are in covering the expenses. So, and we have that, built into the clinical trial as well. So,
Especially if you...
Brian, John.
Yeah, just a brief addendum to that, when we did the type 1 valproic acid study, I was amazed. This is the most highly motivated group of parents that we deal with, and they came from North Carolina up, and they would drive their babies there. So I initially was concerned about that also until we did this type 1 valproic acid study, and I think that, as Jerry said, that Children's does such a good job of accommodating them, and the parents are so highly motivated that I don't think it's going to be an issue.
I have no doubt that the parents would be very highly motivated and are a terrific group of parents. And that's why I want them supported. So my next comment actually referred to that as well. The question of uncompensated harms was disturbing to me. This was my first RAC review. You've probably, 133 times you've heard this, but it was the first for me to really understand that because there's complete unknowability of the effects, even if you did everything right and didn't make a mistake, if there's harm to the patient, then the patients have to sort out the etiology of the harm, and it's not paid for. And it's not paid for by the researcher, by the hospital, and apparently not by the insurance cover, because it's an experimental trial. So they're left being fully responsible for whatever harms are carried by the patient, by their baby. And there was something I thought that was tragic, inherently tragic about that. Again, not particular to your trial.
Well, again, we agree with you 100% on this point, and we went to the head of the IRB. Incredibly agrees with it as well, and this has been a sticky point. We went to the president of the research institute and discussed it with him, and discussed it with the hospital attorneys. And I think there is room for modifying that approach. One of the things that they came back with is a survey that's been done, and then I did my own personal survey with other gene therapists. And I don't have to name the hospitals, but I can tell you there's some very notable programs that have exactly the same built-in limitations on their consent form, and it's really an insurance issue. And I think we can get it modified. It'll take a little work, but we agree entirely. The fact that the IRB agrees and the head of the IRB agrees is a real advantage, and if you put it in your report, that'll further strengthen our approach. So I'm appalled by that as well.
Thank you. I mean, I did use strong language there. I think I turned it Kafka-esque, that there was no way out of this dilemma. They're asking the patients to bear totally all the risk, all the travel, everything, and there was no way to compensate for harm in a high-risk situation. So if we could make that a recommendation from the committee that this be looked at. It's
I
a
will
very
say
small
this is
N.
my third appearance at RAC, and nobody's ever brought it up before, just as a comment. But
That's
and
when you get-
we
right.
weren't expecting it, but
Right.
I think I agree. I mean, I've been appalled by this for a long time, and I agree 100%.
Well, I'm new and I'm a justice theorist, so you're going to hear a lot about this every time. And I'm glad that the senior leadership did get those comments and will think about it, especially here with a very small N and very, very severely affected patient population and parents. And as you said, very, very dedicated parents. This would be, I think, time to either make an exception or change the policy entirely.
Thank you.
So you satisfied with all the answers?
I'm satisfied with the answers and wish you Godspeed.
Very good.
Thank you.
Again, let me also commend you, Dr. Mendell, and the team for all the work that you put into this. This is a wonderful protocol, and I wish you all the best of luck with making progress in this area. I have a couple of questions, and first, I know that the AAV will go through the blood-brain barrier early in life, and that's one of the reasons for choosing this vector. But from your primate and pig studies, you must have a idea what the differential expression would be within the target cells by giving it intrathecally. I'm just wondering, because looking at your data, it's all, it's about getting the gene there and getting it expressed and getting the function. And right now you're running up against a limit of production at probably at about 3.3 times 10 to the 14th per kilo. And that's probably as high as you're going to be able to go. But lower than that, you've tested in animals, it really has not produced great long sustained effects. So it's about getting the gene there, and so would intrathecal delivery make any difference? And then my second question that's related is, what is the window within which you can redose and have it effective in terms of delivering more genes, both intravenous and intrathecal? And the reason I ask is because one of the reviewers here brought up about the lower doses, the first cohort and possibly the second cohort, and whether they are to expect any biologic effect at all in terms of benefit. And so in this uniformly lethal disease, one other way to get around it is to say we're going to go with a low dose for safety and then redose that patient once we know that that dose is safe, and do it as a repeated dosing kind of scheme. And so I'm just wondering, for me, what the window is for intravenous and the window is for intrathecal that you can redeliver vector and have the gene get to its target.
So you certainly bring up a great amount of data that we could probably spend at least half an hour on, but I will be brief.
Yes.
Our systemic gene delivery program was the first, that we had the first discovery within my laboratory. Kevin Faust discovered that AAV9 could cross the blood-brain barrier, and we rapidly moved forward with our systemic gene delivery studies that you saw in the preclinical data and the safety studies that have been presented here as part of this program. The first question is, intrathecal, is it going to be better? Is it going to be the same? First, we don't know the answer. What I can say, if we utilize a GFP vector, and we're only paying attention to the spinal cord, one can target motor neurons at all time points that we have done to date, and this is going into older, our own studies in mouse treated early, one can target motor neurons. As one moves into non-human primates and larger species, it appears that one can target motor neurons at any time point tested, including 1+ years. That's targeting motor neurons, and I'm saying that targeting motor neurons is relatively the same via vascular delivery versus intrathecal. Certainly intrathecal does take less virus to target the same percentages of motor neurons. What I can tell you from our preclinical data is that the SMA mice develop what appears to be an autonomic nervous system disorder, and it manifests itself as far as a dilated cardiomyopathy. We have published this in Human Molecular Genetics in 2011. The mouse certainly shows these autonomic issues, autonomic nervous system issues in SMA patients, in particular type 1 patients. There certainly is